Hello guys and welcome back to Excelsior Academy. So we were doing cell structure and function. So in the previous lecture we have talked about the NPC, nuclear pore complex, right? And we have seen like how in electron micrograph the NPC will look like. Then what are the different proteins which is helping in maintaining the cytoskeleton structure or the entire nucleus which is holding up in the 3D form, right? Now let's discuss more in detail about the NPC, the nuclear pore complex. So these nuclear pore complex are the only channels through which any polar molecules or ions, including your macromolecules like proteins and RNAs, will travel between the nucleoplasm and the cytoplasm. Okay, so as I mentioned before that there are certain proteins, okay, like exportins, then importins are there. So all these proteins are helping in the movement of these molecules. Now NP is an extremely large structure, around 120 million Daltons in humans, 30 times the size of a ribosome. So अगर ribosome इतना है तो आपका NPC इतना होगा, ठीक है? Now invertebrates in the nuclear pore complex is composed of multiple copies known as nucleoporins or NUPs. Okay. By controlling the traffic of molecules between the nucleus and the cytoplasm, the NPC plays fundamental role in physiology of all the eukaryotic cell. Now, why it is helping in making the like how the controlling of the traffic of molecules is maintained and why it is needed, so that depending on the environment stimulus, the gene expression can be happen. Okay, like whether you need to have the genes to be shut off or shut on, so that can be regulated. Now same thing here, what we are saying is the RNAs which is synthesized in the nucleus, they should be transported to the cytoplasm, और जो proteins हैं, जो enzymes हैं जो आपके cytoplasm में synthesize होते हैं, for your transcription you need it, for DNA replication you need जो आपका nucleoplasm में होता है, right? So all these enzymes after synthesis in their functional form should be transported back to the nucleus where they can carry out the DNA replication transcription process. Okay. Now, this is just the another figure just to show like how molecular trafficking is being held. So passive diffusion where the small molecules, ions can travel. Selected transport of the proteins or RNAs depending on the requirement. Like RNA आपको मालूम है, mRNA should be taken out, right? tRNA should be taken out. Ribosomes should be taken out in the cytoplasm so that the translation can happen. Then after the translation, the proteins, if they are acting as a DNA polymerases, then they should take inside of the nucleoplasm, right? So this is just the summary of that. Now this is the electron micrograph and nuclear pore complex. So last time what image we saw was the not the of high magnification. तो हमने क्या देखा था, ऐसे ऐसे depressions were present and those were NPC. Now this is a closed view of your NPC. Now what do we see here? That these NPCs, they look like a flower like structures, right? Those are nothing but the number of helical integral proteins, of which like eight are present. I don't know, 1 2 3 4 5 6 7 8. So like this, if you see like flower like structure is present, 8 structural subunits are present, and in this portion is the pore from which the molecules are getting transported. Okay guys, are you with me, able to follow?
Yes, ma'am.
Okay, now coming to the detailed structures. So here, see, this is again, as I said, it is made up of 8 subunits. तो हमने यहाँ पे half लिया है, 4 का लिया हुआ है, ठीक है? This is like the transverse section of your protein. Sorry. Now, these are your cytoplasmic filaments. Okay. Now these filaments are actually helping in interactions with the proteins. जैसे importin हो गया, exportin हो गया, right? So ये वहाँ पे आपको help करेगा bind करने में. Then you have got the FGNUPs. Those are your, the nucleoporins, which is present over here. Okay. They regulate the transport. Then you have got the nuclear ring, the basket. All these are present just to ensure that all importance exportins are present nearby. And एक और चीज आपको ध्यान रखना है यहाँ पे, that importance हो या exportins हो, they cannot function without energy. सो उसके लिए आपको जीटीपी लगेगा। तो जीटीपी के लिए हमेशा क्या होगा, जीटीपी इज गोइंग टू टेक एंड ट्रांसफर इट, फिर हाइड्रोलिसिस होगा। तो दीज इम्पोर्टिंग्स मतलब क्या, बाहर से अंदर लाना, करेक्ट, बाहर से अंदर लाना। पर ये इम्पोर्टेंस को वापस साइटोप्लाज्म में जाना होगा, so that in future it can bring again the components required in the nucleoplasm। तो उसके लिए आपको जीटीपी लगता है। Same way exportins are present inside the nucleoplasm। ये कोई भी आइटम लेके बाहर जाएगा, ओके। अब इसको भी तो वापस आना है। So all this mechanism needs energy and that is what we have to see, okay। यहाँ पे confusion नहीं होना है। See again, the importins exportins are nothing but proteins only, which is helping in bringing the required transcription factor or RNA near to the NPC so that they can be exported or imported। But at the end of the day, these importins and exportins are also proteins। उनको भी अपनी अपनी जगह पे जाना है। तो ये सब कैसे रेगुलेट होता है वो हम इस सेशन में देखेंगे, ठीक है। So the selective transport of proteins to and from the nucleus। तो हम अभी यहाँ पे proteins का देखेंगे। Guys, you understood the concept over there, like how importins and exportins are doing their job and why ultimately at the end of the day they are also proteins. So even they have to be exported and imported so that they can go to their respective location. Are you able to follow this? Akshaya, clear to you?
Yes, ma'am.
Okay. The exact mechanism we'll study eventually, but this is a just like what we are going to study, okay। Now this is about like what determines the macromolecules to be, you know, brought to the nucleus। abhi dekho, so in this case let's say that you have got, this is your nucleus, okay, ye aapka nucleus hai। now you need proteins which needs to be recruited to the nucleus, ठीक है। तो जैसे your letter, your parcel has got the address, looking at the address only the delivery guy or the postman is understanding, okay मुझे ये particular parcel यहाँ ले जाना है, ठीक है। in the same way the proteins also have that signal। in the case of nucleus it is known as nuclear localization signal, ठीक है, NLS signal। So exam में directly NLS signal लिखा होगा। So from there you should understand that these are the signal which is present in the proteins to be recruited through the NPC inside the nucleoplasm। समझा? ये होगा आपका import करने वाले proteins which have got the NLS। Clear? इतना clear है कि NLS कहाँ present है? On the proteins which you are going to recruit inside of the nucleoplasm, okay। now this is the example like how this NLS looks like। so in the case of the T antigen, so T antigen kahan milta hai aapka Civian virus mein, so in that they have studied first, okay, in the T antigen, later they studied in the nucleoplasm in the eukaryotes, pehle virus mein padha uske baad eukaryotes mein padha। so it is a single stretch of amino acids। NLS of nucleoplasm is bipartite। bipartite matlab uska jo signal hai it is divided in two parts, here and here, ठीक है। now when you say bipartite it is divided in two parts। now why it is important, because it is accordingly going to have interactions with the importance, okay, and then only this importance will bring the nucleoplasm to the nucleus, okay। now let's see what is the signal over here। so mainly in the case of T antigen it is proline, lysine, arginine, lysine, valine। in the case of the nucleoplasm, lysine, arginine and lysine। so overall what we have seen is that the NLS is your basic, it is positively charged, correct? Able to follow? What we see is lysine and arginine is present, so both are positively charged amino acids। when you have got more expression, so your NLS, that is this nuclear localization signal, is positively charged। so iska opposite we are going to expect in the case of your importance. Clear? Is this slide clear to everyone? Tanvi? Tanvi, it is clear to you?
Yes, ma'am.
Okay. Now, so same thing, proteins with classical NLS, they are recognized by the transport proteins called importins because they carry the proteins through the NPC into the nucleus. Okay? And these importins works in conjunction with GTP. So GTP binding protein यहाँ पे RAN है. इससे पहले हमने पढ़ा होगा तो if you remember in the transcription translation whenever we need GTP exchange वहां पे क्या था आपका जेफ था guanine exchange factor. Okay, in the case of nucleus it is RAN. So RAN is also a GTP binding protein. So the conformation and activities of these proteins are regulated when they are bound to GTP and GDP. So normally हमने क्या देखा है when it is bound to GTP and hydrolysis होता है there is a change in the conformational or the structural change which results in the opening of the pores. Correct? Yeah, you know the structural change which will bring about the opening of the pores so that the transport of molecules can happen. Same way here also it is because of the binding of GTP and affinity towards the GTP and GDP. Okay. Now, one more thing here extra is that the high concentration of RAN GTP in the nucleus will determine the direction of nuclear transport. ठीक है? तो एक reason हो गया that NLS will be present on the proteins which will be recruited inside of the nucleus. Same way you have got the exportin signals also. यानी कि nucleus से बाहर ले जाना है. Okay, so ये सारे signal and as well as the concentration of your RAN GTP is going to decide in which direction the transport is going to happen. Okay, now let's see the protein import through the NPC. Okay, so first thing is look at the first figure. Now, what do we see here? That you have got a cargo protein. Let it be any protein. NLS is there. To this NLS, the important protein is binding. Okay. When this goes and binds, it goes and make interactions with these cytoplasmic filaments. Then, when it is understood that this is correct, the nucleoporins proteins will allow it to pass through the NPC, where importin and the cargo protein will dissociate. Clear? Clear? Is it clear? This part? Itna clear hai?
Yes, ma'am.
Okay. Now, importin आपका हमेशा cytoplasm में होना चाहिए. Correct? Then only it can function. So right now, we need to take importin out of the nucleus to the cytoplasm. So this RAN GTP, okay, GTP will come and bind to this importin. So now importin bound with the RAN GTP, this one will be carried outside where the dissociation happen. यानी कि क्या हुआ RAN GTP hydrolysis हुआ. Okay, with the help of RAN GAP. Now importin आपका बाहर आ गया. Correct? Now this RAN GDP is there. Okay, able to follow? This is second part of your nuclear transport. This is first part of your nuclear transport. तो पहला क्या था इशू कि आपका इम्पोर्टेंट अंदर आ गया था, अब हमने उसको बाहर ले आया है. ठीक है, so that now it is ready for another protein to bind and bring inside the nucleus. Okay, अब मुझे RAN GDP अंदर लाना है, for which I'm gonna bind to another protein called N2F2. NTF2, sorry. Okay, this NTF2 will bind to the RAN GTP which will be bring together out inside. अब मेरा RAN GDP जो है with the help of the JEF it will get exchanged to the GTP and then again that cycle will continue. Clear guys? Did you able to follow the all the three part? Any part which is not
Yes,
clear?
ma'am.
Huh.
The NFT is now inside, right? It also
Huh.
has to be taken out.
So this is actually your import receptor. ओके सो दिस
Yeah.
हाँ सो ये साइकिल आपका ऐसे ही चलता रहेगा, अंदर बाहर, मेकिंग श्योर दैट एवरीथिंग इज देन इन प्लेस, सारे काम होते जाएंगे. So that's why we say na, even though we sleep at night but our cells don't, because all these transport is still going on.
No, ma'am, I mean NFT can't work unless it's outside as well, right? So
So
now that NFT is
NT
inside, it has to go.
हाँ,
Sorry, NT, yes.
so NTF2 again, so it will be bound to other proteins to be taken out. ये जैसे कि NTF2 है, right? So this will be should be present outside, correct? This is what you are saying, है ना Shruti?
Yeah.
हाँ, so this NTF2 will have its export का sequence. This will bind to the exportin proteins and it will be taken out.
Okay, okay.
हाँ, ये एक part of the NPC हुआ है. Okay, clear? So I'm not going through all this what we have written here. It is the same thing which I explained. Okay, so पहला I'll just summarize it again. Importin goes and binds to the annulus part of your cargo protein. It goes inside. It is released. ये अपना काम करने गया gene expression के लिए. Importin needs to be taken out. So it goes and binds to the RAN GTP. Okay. Then goes outside, hydrolysis loses affinity for the importin and becomes GDP. और ये hydrolysis activate करता है आपका RAN gap protein. Okay. Now this RAN GDP should be taken inside, which is done with the help of the NTF2. NTF2 वापस ले आएगा, RAN GDP GTP बनेगा with the help of the JEF. Gone. Now NTF2 will go and bind to the exportin proteins. ये आपको बाहर भेज देगा. And the cycle continues again and again. Yes, clear now?
Yes, ma'am.
For everyone, Akshay and Tanvi?
Yes, ma'am.
परेशान मत होना क्योंकि मैं 10 बार पूछ रही हूँ कि क्लियर है कि नहीं, बिकॉज सी जब हम when you guys are going to do MCQ, this all is required step by step. Remember how I explained before, like जब कोई भी ट्रांसपोर्टर होता है, कोई भी चीज बाइंड करता है, तो पहले क्या होता है, इट हैज टू बी स्टेप वाइज, करेक्ट? So here it is, apart from understanding like how the transport is happening, it is also required to know कि कौन सा step पहले हो रहा है, because it is highly regulated. So till now for the with respect to your exam we study that ये यहाँ आता है, ये ऐसे bind करता है, ये ऐसे होता है, but no, in reality the binding of this importin and the protein to the cytoplasmic factors involves many steps. Okay, but which is at the research level now. So ऐसा नहीं है, so every step is highly regulated. Okay, and that's why the step matters here. It's not the output but the steps which is matting which is very important in this case. Okay, it matters a lot. Okay, now like how we have got the nuclear localization signals, export signals are also there within the nucleus called exportins. Okay, which will go and do the transport. So they also have got the family. So here we have got these. So these are known as carioferins. Okay, please remember. These are known as the carioferins, the import proteins and the export proteins. So importin with basic amino acid is NLS. SNR protein यानी कि SNF के लिए. Transportin for mRNA binding proteins, ribosomal proteins. Importin 7 for histone H1 ribosomal proteins. ठीक है? Import करने के लिए. अभी देखो ये सब याद भी करना है. इम्पोर्टेंट 7 का काम है सिर्फ हिस्टोन प्रोटीन्स लाना, राइबोसोमल प्रोटीन्स लाना. देन ट्रांसपोर्टिन का काम है सिर्फ एमआरएनए बाइंडिंग प्रोटीन्स, इफ यू रिमेंबर, राइट? एमआरएनए को पहले हमें उसको एक तो सिंगल स्ट्रैंडेड रखना है एंड फिर उसको एमआरएनए को आपको बाहर साइटोप्लाज्म में लाना है, so that the translation can happen. Okay, similarly we have got for the export also. So proteins with leucine rich nuclear export signals and all these things are taken again back, exported out by the help of the CRM1 proteins. Then exportin T, exportin 5 for your tRNAs, miRNAs, etc. Okay, this is the still the basic common import and export proteins, but we have got more than 100 and 200 proteins, which is they are doing the function. So in short, you need to understand that हर एक कैटेगरी ऑफ ट्रांसक्रिप्शन फैक्टर को अगर मुझे न्यूक्लियस में लाना है फॉर जीन एक्सप्रेशन, आई हैव गॉट द स्पेसिफिक इंपॉर्टेंट प्रोटीन. ऐसा नहीं कि कोई भी इंपॉर्टेंट प्रोटीन लेकर आ सकता है, ठीक है. वी हैव गॉट specific proteins each aligned or each assigned for bringing the respective transcription factors or any other proteins involved to inside of the cell. Okay. For example, let us say NF kappa B is a transcription factor. DNA polymerase is your enzyme, correct? But at the end, they both are protein. So we have got different important proteins to bring these two different proteins inside the nucleus. Clear? Now, same here, like how we have discussed about the RAN GTP involvement with the importance, same way we have got for the exportins also. So exportin binds to RAN required for nuclear export as well as for nuclear import. So RAN GTP promotes the formation of stable complex between the exportins and the cargo proteins, whereas it dissociates the complex between the importins and their cargos. Okay. This effect of RAN GTP binding on exportins dictates the movement of proteins containing the nuclear export signals from the nucleus to the cytoplasm. So exportins, they form stable complex with the cargo proteins in association with GTP within the nucleus. इसका मतलब क्या? अगर RAN GTP हाई एफिनिटी है तो एक्सपोर्ट प्रोटीन्स के साथ है, ठीक है. और अगर लो एफिनिटी है तो वो इंपोर्ट प्रोटीन्स के साथ है. समझ में आया? क्लियर? द समरी ऑफ दिस स्लाइड इज, if suppose this is the nucleus, not like this, sorry, this is the nucleus, ये आपका NPC है, let us say. okay, import proteins कहाँ होने चाहिए आपके? साइटोप्लाज्म में, करेक्ट. So the association of your RAN GTP and importance will be less, यानी कि importance and RAN GDP होगा. So here the dissociation rate is high, and inside here you are going to have more with the export proteins. समझ रहे हैं? Yes. I wanted to hear from you guys that whether are you able to follow or not.
Yes, ma'am.
Yeah, okay, good. So following this transport to the cytosolic side of the nuclear envelope, the hydrolysis and the release of GDP leads to dissociation. Okay. Now exportins as well as RAN GDP, they are then recycled to the nuclear pore complex for the reuse. So this we all have seen, right? Now let's see the same thing with the help of nuclear export. Okay, now look at the figure. So again, cytoplasm, nucleus over here. Okay. Now, see the cargo proteins बाहर आ गया, आपका GDP भी बाहर आ गया, right? Now what happens is that this exportin will go and bind. Can you see NTF2? वो भी उसके साथ ही है. So all what you see is that every time this import export आपका चलता ही रहेगा. ठीक है? Do you want me to explain this figure? Or it is self-explanatory? Yes, should I explain? Okay, Shruti, would you like to try explaining this figure?
Okay.
I'll tell you, start from here.
And I'll be starting from the cargo protein part.
Like
so,
since it,
sorry?
No, I was saying that since we are talking about export, so start from here, saying that cargo
Okay.
protein binds to the nuclear export signal, etc., etc. Yes.
Okay. So, like you said, the cargo protein first binds the nuclear export signal so that it can find the nuclear pore, and then it gets out. of the nuclear pore. And there the cargo protein dissociates from the export protein, which is exportin. And then that has to be, exportin has to be bound to RAN GDP so that it can get out. And then when it's once it's outside, RAN GDP is dissociated. And then exportin goes toward, nuclear pore. It is recycled back through the nuclear pore. So exportin has to bind to NPF too. And RAN GTP has turned into GDP right now. So it also binds to RAN GDP and then goes back in through the nuclear pore, where it again dissociates. And then RAN GDP and NPF are again, they are still together, so they also dissociate. And then the RAN GDP gets
exchange,
becomes RAN GDP through RAN GEF.
yes,
and then the cycle continues.
Yes. In short, whenever you do export import, RAN GTP is required. Okay. See, यहाँ पे भी GTP. So inside of the nucleus, it is GTP which is helping in the transport from nucleus to the cytoplasm. Now, in another words, question depend करता है, ठीक है? In another words, you can say that it is RAN GDP in the cytoplasm which brings your exportin to go inside in this direction from cytoplasm to nucleoplasm. Samjha the difference? So here in the exportin we need GDP association, whereas in the case of here in the case of export out करने के लिए मुझे चाहिए GTP. कोई भी चीज न्यूक्लियस से साइटोप्लाज्म जा रहा है, it is GTP. Or in other words, if anything is coming from cytoplasm to nucleus, it is GDP. Simple. ठीक है? इसी पे based question आएगा. To confuse you guys. तो फ्लो चार्ट ऐसा बनाओ या अंडरस्टैंडिंग ऐसी रखो that nucleus to cytoplasm it is GTP, cytoplasm to nucleus it is GDP. ठीक है? So in the case of nucleus it is RAN. जब हम साइटोप्लाज्म में जो जीटीपी की बात करते हैं वो रैस होता है. ये रैस जीटीपी बाइंडिंग प्रोटीन है. इन द केस ऑफ न्यूक्लियस इट इज रैन. ओके, there are also many different types of GTP binding proteins are there. Okay, clear?
Yes, ma'am.
Okay. Oh yeah, so this much only I prepared. I just want to take yeah, I'll just stop